Good morning, good afternoon, and good evening, everyone, and welcome to the Scientist.com and GemPharmatech webinar titled Applications of Humanized Mice in the Evaluation of Antibody-Drug Conjugates to Treat Autoimmune Diseases. I'm Andrew Davies from the events team here at Scientist.com, and I'll be your host today. We're very fortunate to be joined by Dr. Jun Wang, Director of Scientific Engagement at GemPharmatech, and Dr. Chen, President of BioRay Pharmaceutical, who will discuss the use of humanized mice to evaluate antibody-drug conjugates for treating autoimmune diseases. And without further ado, it is now my pleasure to welcome our first speaker, Dr. Jun Wang. Jun, welcome, and please take it away whenever you're ready.
All right, thank you for the introduction, Andrew. It's my pleasure to be here and to present some of the work GemPharmatech has been doing in the past few years. And hopefully by the end of the presentation, you can learn some of the new models we created to advance the development of novel therapeutic antibodies and antibody-drug conjugates. For those who do not know GemPharmatech, we are a global biotech tech company specialized in making genetically engineered mouse models. We currently have the world's leading mouse model repository with over 25,000 models in stock. And we continue to make new models every year with the capacity of generating over 6,000 new models. We also offer preclinical testing services using these innovative models. And as we know, there's a limitation of using conventional animal models for antibody-based therapeutics, including antibody-drug conjugates. There are several reasons. For example, the therapeutic antibodies often have limited cross-species reactivity. So these antibodies can only bind to certain species, for example, human target, but oftentimes not mouse target. So this creates some difficulty to select the right models. And also certain drug targets are only expressed in humans and other species, but not in mouse. But we have a way to overcome these limitations. And third is whenever we work with mouse models, especially the immunocompetent models, they only have mouse immune system, but not human immune system. So there are some differences between the immune system of mouse and human that make the translatability of the research sometimes limited. We have a few strategies within GemPharmatech to try to overcome the limitations of conventional mouse models. So today I'm going to cover two strategies we have used. The first strategy is so-called human gene knocking models, also known as gene humanized models, in which we will introduce the human gene into the mouse genome. And the second strategy is we are using human immune system reconstitution model in immunodeficient background. And the last part I'll be talking about a little bit of the resources we created to accelerate autoimmune disease research. This is a summary of how we usually generate humanized models, especially the gene humanization part. We can either introduce the coding sequence of a human gene into the mouse genome, so the end product will be a whole gene humanization. The whole protein will be human. And typically we insert the human gene into the endogenous mouse gene locus. The benefit is the gene expression is still under the control of the mouse endogenous promoter. So tissue expression specificity, expression level will be very similar to the mouse endogenous gene. Another strategy is called partial humanization. This strategy is very, very typical for example, transmembrane protein, because we want to humanize the extracellular part of the protein to engage therapeutic antibodies, and the intracellular domain will still remain as mouse, because we do want to keep signal transduction intact by interacting with cytoplasmic mouse proteins. By using these strategies, we were able to generate a big library of these human gene knocking mouse. And not only we were able to generate single target knocking, but also double, triple, sometimes even quadruple targets. And we oftentimes make these knocking models both in C57 Black 6 background and above C. Here, I wanted to just use a few case studies to show how we typically use these humanized models to evaluate therapeutic antibodies in a humanized mouse setting. So in this case, we are using a humanized TSLP, TSLP receptor double knocking mouse in atopic dermatitis setting. So this is a typical study design in which we would induce the disease onset using MC903, which is a vitamin D analog. And then we used a positive control drug, tezepelumab, which is an anti-TSLP monoclonal antibody to evaluate whether we can see reversal of the phenotype in this setting. As you can see here, whenever we treat the mice with MC903, we can observe increased skin thickness and clinical score, as well as increased lymph node weight, serum IgE production, and the scratching frequency. And when these mice were treated with anti-TSLP monoclonal antibody, we can see a reversal of the phenotype. So there's a reduced skin thickness, clinical score, and other phenotype in this model. We also check the cytokine expression in the skin. What we can see is there's a mixed Th1 and Th2 immune response triggered by the MC903 treatment, and the anti-TSLP monoclonal antibody was able to reverse some of the phenotype, including reducing the levels of inflammatory cytokines, Th1, Th2, in these skin sites. And then in another case study, I wanted to highlight the use of humanized PD-1 mice. So the disease setting in this case, we are using a mouse EAE model induced by the MOC peptide in combination with CFA treatment. So we tried the anti-human PD-1 antibody, which is an agonist, to evaluate whether this model will behave, how the mouse model will behave. And as we can see, we used the EAE score to compare to evaluate the efficacy of the PD-1 agonist. So rosnilimab is a clinical stage PD-1 agonist. And by using different treatment regimen, either to start early treatment at day 0 versus later treatment at day 10, we were able to see starting early treatment will reduce the both the average EAE score and the cumulative EAE score in the mouse EAE model. Here, fingolimab is a sphingosine 1-phosphate receptor modulator. Here, it's an approved drug that we use here as a positive control. So I just talked about the human gene knocking models. The second strategy I'm going to cover today is the human immune system reconstitution model. I have to introduce the NCG line, which is the foundation of our severe immunodeficient mouse portfolio. So the NCG mouse is immunodeficient. It's lacking T cell, B cell, and NK cells. Built on top of the NCG, we generated over 40 different next generation NCG lines. These NCG models often express different human cytokines or human drug targets to really further facilitate the use of the NCG model. And we created a wide range of different NCG derivatives for the purpose of supporting human immune system reconstitution. And based on what type of human immune cells you wanted to reconstitute, we would make different recommendations. So in these cases, the NCGN model, which overexpress human SCF, GM, CSF, and L3, these models we select because they can reconstitute a wide range of different human immune cell types, including human myeloid cells. And oftentimes, the induction of the autoimmune diseases involve many different immune cell types. So this is a very suitable model to model human autoimmune diseases. And here we compare the NCGM immune cell reconstitution versus the parental NCG line. And we can see compared to the original NCG, which really reconstitutes the majority of human T cells, the NCGM was able to reconstitute a more diverse human immune cell types, and there's a higher level of human leukocytes that can be reconstituted in the NCGM. And furthermore, we can also see NCGM can support the reconstitution of different human myeloid cells significantly better than the parental NCG. So using these NCGM as a foundation, we were able to test a few chemically induced models using the reconstituted NCGM. So typically we would inject the human HSC into the NCGM after irradiation. So after about 12 to 14 weeks, different human immune cells will be reconstituted. At that time, we would inject CFA into the subcutaneously into the foot pad of the mouse to induce the arthritis. So in this case, and you can see when we inject the CFA into the mouse, there's a swelling in the paw, and this mimics the arthritis condition. And when we evaluate the arthritis score, you can see there's an increase of our arthritis score in the CFA-treated mice. And this is a typical study design. We first will reconstitute the NCGM mice with the human HSC, then inject the CFA, then we can evaluate the efficacy of drug candidates, and we have some endpoint analysis. And here we are using the monoclonal antibody against TNF-alpha, adalimumab, as a positive control. And you can see treatment of adalimumab can effectively reduce the arthritis score in this arthritis model. Additionally, we saw a decrease of the paw thickness in the TNF-alpha blocking antibody-treated mice, as well as a decrease of serum IL-6 production in TNF-alpha monoclonal antibody-treated mice. In another case study here, we are trying to demonstrate the use of the same NCGM mice, but in a different disease setting. So here, this is a SLE lupus model induced by TLR7. And here, the reconstitution is, the timeline of the reconstitution is very similar. After the reconstitution, we would induce the disease onset using the toll-like receptor 7 agonist to induce a lupus condition. Then following that, we can start efficacy study and evaluate therapeutics against lupus. This is some of the data we generated. We can see that after the TLR7 agonist induction, there is a increased percentage of human CD4 positive cells and activated T cells. And also we observed activated PDC cells, as well as increased serum IgG level and anti-double-stranded DNA level. Next, we tried to use a few B-cell targeting antibodies and see whether there's any effect in these humanized lupus model. And you can see compared to the control group, which is the group 2, there's an effective reduction of the anti-double-stranded DNA and IgG, specifically in the CD3, CD19 bispecific antibody treatment group. So this is a T-cell engager that targets the CD19 in the B cells. We were also able to see the depletion of B cells. So if you look at the flow analysis on the human CD19 positive B cell population in group 3 and group 5, there is a big reduction of B cell population in these T cell engagers CD19 bispecific antibody treated groups. And I hope by now I was able to show some of these animal models, humanized animal models that can be helpful to evaluate some of the antibody therapeutics. In addition, we actually have a large resource to help with autoimmune disease research. This is a summary slide I wanted to show that shows all the different a big collection of autoimmune disease models we have. Some of these are genetically engineered model indicated in the red color. So these are spontaneous models, and other models, including the ones I talked about today, these are chemically induced models. So the benefit of using chemically induced models are we can easily apply the same study protocol on the humanized background, the gene humanized background. So in these cases, we were able to evaluate the drug efficacy if they don't interact with, if they have limited cross-species, if they cannot bind to the mouse antigen. And not all the models are made the same. So using this as using the SLE models as an example, we built different models based on the mechanism of action of the drug. And we can make different recommendations because some of the models will be more suitable for evaluating certain drugs, but not the others. So we typically would list very clearly which model is better to test which drug based on the mechanism of action. And finally, I wanted to also highlight a recent product we just launched, which is a fully human antibody producing mouse. As we know, antibody is a critical part of the antibody-drug conjugates. So having a new mouse model that is able to produce fully human antibody can really minimize the humanization process and accelerate discovery of the therapeutic antibody and the ADC. With that, I would like to summarize the talk. So we know, I hope by the end of the talk, I was able to convince you we are able to model autoimmune diseases using different types of humanized model, and these can really overcome some of the limitations of the conventional models. And second, we also wanted to let you know it is critical to select relevant disease models, depending on the mechanism of action of the drug candidates. And the last one, lastly, we wanted to say the immune system is very complex, and we continue to develop new models and optimize these models. So it is very necessary to continue the innovation of these animal models to generate more clinically relevant animal models. And with that, I wanted to give the stage back to Andrew.
Wonderful. Thank you very much, Jun, for that great presentation. I really appreciate it. And now it's my pleasure to welcome Dr. Gong Chen. Gong, we'll bring you back on here. Welcome, Gong. Please bring up your slides and take it away whenever you're ready. As Dr. Chen pulls up his slides, just a reminder for everyone that you can submit questions at any time using the Q&A panel, and we'll answer as many as we can in a Q&A session at the end of the presentation. All right, take it away, Gong.
Yeah, great. Thank you, Andrew, for the kind introduction. Good morning, everyone, and thank you for joining us today. I'm really excited to introduce our BioRay our IL-4R ADC program. This is a good collaboration with Genentech. As you say, it's very important to find the right model to evaluate a drug, especially for the immunology disease, which is just so complicated in terms of the different pathway can cause the underlying immune cause. So today, before I dive into the more detailed for our ADC program, I'd like to first introduce our company. So BioRay is a commercial state biopharm headquartered in China. We focus on the oncology and also immunodisease. So this we are the number one in terms of the sales for the autoimmune disease product in China. We have a total of 6 commercial products on the market. 5 of them is oncology, immunodisease related, and 2 is anti-cancer drugs. So last year we generated 200 million sales, and this year we expect to generate about 300 million sales, which is really high. The growth rate, which is much higher in our industry in China. And we have also, besides those 6 products, we also have multiple pipeline in oncology and autoimmune disease. So today I'm going to focus on more the autoimmune disease pipeline. So we have about 2,000 scientists and engineering and production people, and so it's just a pretty sized biopharm company in China. So as I mentioned, we are focused on the immunodisease. And so this is the platform we built for the autoimmune disease platform. So we have developed 3 different technology platform. One is an antibody, which is a monoclonal bispecific antibody and a biparatopic antibody we have developed. And also with that, we have called the ADC, which is focused on immunology disease. And also we have a T-cell engager, which is a bispecific trispecific antibody, which is use T-cell as a target, as the depleter. So for those, the most advanced target product we have is the BDCA2. So we actually take a 2-pronged approach, one using the monoclonal approach. The other one, we also do using the IMAT ADC approach to target the BDCA2. The one that we've talked about today is the IL-4R ADC. I'm going to give it more detail on that one. So for our BioRay ADC, we have the full capacity and full expertise. So we can have an antibody from discovery to the GMP manufacture of antibody. And we also have a bispecific and biparatopic antibody for low expression target. Our BR111 is the biparatopic antibody against ROR1 ADC. So this ADC we are expecting the clinical early next year. And also we have an array of the payloads, especially for oncology. We have the microtubule inhibitor and topo-I inhibitor. And recently we also find some other HDAC and other payload that can use for ADC. And most importantly, we also have a bio-ADC platform which can combine those 2 payload in a 1 antibody ADC. So that one we expect to have an IND next year for BR116 program. Also, beside those standard payload for oncology program, we also have a variety of immunomodulator agent for ADC for those purpose. So we have for agonists for immune oncology project, and also for antagonists for immune-related disease. So with those payload and an antibody, we also develop a cis-link and cis-X conjugation method. I'm going to focus more, give you more detail about our cis-X conjugation. It's a conjugation that cross-link the 4 disulfide bond in the antibody in the hinge region. It's irreversible. compared to the other current industry used conjugate method. So the cis-X is irreversible conjugation, and the conjugation site is very stable. And the site also specific only for those 4 cysteine site. And DAR is very precise for DAR4. And the antibody, because they cross-link the disulfide bond, so antibodies itself is very stable, and the whole entire ADC, entire antibody. So integrity is reserved. And the very important is for the CMC, especially for the commercial stage, is the manufacturer. This conjugation method is a one step, one part, and there's no enzyme. You don't need to remove the enzyme, and can scale up. So our BR111 ADC actually is using this first one using this cis-X conjugation methods. So regarding to the ADCs have been developed almost 20, 30 years. So most of the targets against the oncology. So ADC for immunodisease is relatively the kind of early stage, but I think it's there's more and more interest using ADC for the against the autoimmune disease. So this is some example of early stage the people try to make ADC for autoimmune disease. I think the most successful ones, FV, they have the this bottom one is TNF ADC. They're using TNF as an antibody, anti-TNF antibody as the target agent. So this AB3373 is advanced to they finished the phase 1 trial, and they see some encouraging clinical results. So they actually modify this one to improve the solubility, like they improved add a sulfate group into the payload to increase the solubility. They tried this one. Unfortunately, a few years ago they terminated this project. I think it's because they didn't see a significant the benefit versus the antibody. So, but at least it's pay the role for industry, for us to move this direction. So one thing that the reason I think is this antibodies ADC TNF ADC is not working is probably the targeting part, because the TNF itself is not good targeting the ADC target, so its internalization is relatively low. So that's the reason kind of we are thinking that we need to find a good targeting for the ADC. So R4R1 is automatic. It's a good ADC is a good target for ADC. So that's where we are doing the anti-IL4R ADC for atopic dermatitis. So atopic dermatitis is a chronic inflammatory skin disease. It's by the allergen, it's caused by genetic and also by the environmental factors. So in the US alone, it's about 20 million people affected by this one, and half is children, young age children, and 3 million is have a moderate and severe AD, which is needed interfered by doctor. So, and also this disease is affect more women than men. So not only like this disease affect your daily life, people's life, but also it's a big financial burden for other people and their family. So it's a very unmet need on this field. And also globally, it's about 200 million people affected by this disease. And like I said, it's in general, it's half is children, but also more preferentially is more to the female than the male. So it's just a very big disease and big area to treat. So if you look at the progression of the development of atopic dermatitis, it's infected by genetic and also by environmental. So when the beginning, early stage phase, then the phase 0 and to phase 1, which is caused by all different infection by the bacteria. So the phase 1 really quick advance to phase 2, And this phase 2 that activate the T cell and also the release of the IgE, which is sensitized. So then advanced disease really quick advance to phase 3, which is depend on the which T cell the helperphage effect. So it could become AD and or become the crisis or different disease. So and this from phase 2 and phase 3 is the area which we can targeting to treat to prevent that disease progression. And so that's so the Th2 cell is the really good target to treat on the AD disease. And this is more detailed how in terms of the Th2 cell and also IL-4 get involved. On the right side, you can see there's a disease model on the skin that you get a different kind of environmental and allergen. So activity, the different the skin cell release because in fact the allergen so it release the different kind of cytokine and the cytokine and also affect the DC cell advance to the in the middle that advance to the thyroid. So that one in there the mature the Th0 cell to Th2 and then release the activate the Th2 cell, then Th2 cell further activate the B cell, then release the IgE. So in here, the Th2 cell is very critical, key the cell to targeting, and IL-4 is the one which is activate the T cell and also which itself can self-regulate the cell. So we are targeting this IL-4 cell, IL-4 targeting for the Th2 cell. So then Th2 cell once it mature in the lymph node, then they will advance to the bloodstream and go to also come back to the infection site and inflammation site. So there they're going to further inflame the infection and inflammation. So if you targeted IL-4 targets and which is target Th2 cell, then you not only tune down the Th2 cell, but also when the Th2 cell get to the inflammation side, the release of the small molecule or release the drug, you can reduce the inflammation side. So currently, the three major treatment for the AD is the biologic, and here an example is dupilumab, which is target the IL-4R antibody, and also there's many different JAK and TYK inhibitor, which can target the downstream pathway of IL-4 and other unrelated pathway. And also they use a very common use of the steroid, which is on the top, usually is for on the topic use. So all those treatments still they have room to improve, and also there's actually clinical evidence that the combination of those three, those biologic and small molecule treatment can combine, I think, more effective. And so this is the clinical result of the dupilumab for the treatment. So it's about 40% improvement over the placebo. I didn't show the result. It's they also did conduct the trial with the combo with the steroid, and the activity, the recovery, the potency, the endpoint actually get improved to 20% more. So this also give us the suggestion or indication that ADC might be better. Not only they can improve the potency, but also can reduce the JAK inhibitor or steroid, the side effect. For example, the JAK is a black bar warning. So it's for any kind of chronic disease, long-term use is not really have to be cautious. And also the steroid we can cause the bone loss if you do long-term use. But if you do make an ADC, which can reduce those toxicity or side effect, but at the same time improve the potency for antibody. And so this is the kind of like we are rationale. So for the antibody, we can inhibit the IL-4R, which is the two signal. But for the JAK inhibitor or steroid can inhibit downstream, which is maybe related to R4R or maybe unrelated to R4R. So that's the kind of rationale we have when we start this project. So for the screener R4R antibody, so like I mentioned before, the most important one is the targeting, right? You had to, like TNF for the problem we have. So we want to make sure that the alpha antibody can be highly affinity, very specific, and also have a really high, good internalization rate, so that way you can bring all the payload to the inside the TH cell. And also for those small molecule, like most the steroid and also the JAK inhibitor, they all have a really hydrophobic, the core. So in order to bring those in, you have to develop the linker, which can reduce the hydrophobicity of those the overall the ADC. And the last one is the payload screening. So like I mentioned before, there's a JAK inhibitor and also steroid to use, and also there's like a toll-like or sting-like the modulator, which can be considered to use. So we actually screen all in our lab. We actually screen all those different type of payload on this project. And so this is the one example we screen for antibody. And like you see here is an antibody 1, antibody 2. I can see I have a different internalization rate and also the maximum amount of getting the cell. And also we found is IgG1 and IgG2, a different type IgG other type also have effect in ADC, affect the antibody internal rate. So so it's very important to find a good antibody that can internalize. And so we have actually found by this kind of screening, we find a good internalization rate for an antibody. And then we want to see whether, the antibody does, affect the T cell production, the T cell proliferation. So we use the UHC 9737, which is a T cell type of cell line. And you can see the antibody 1, antibody 2 have a pretty good inhibition. And also you see the payload 1, actually, you can see this have a less potent, but one important thing that see here is the blue line is actually have a more suppression compared to the antibody for the red line. So also you can see when you combine the physical mixture, the antibody with the payload, you can see have a more suppression. So that means to give us an indication that ADC might have a better way to then compare the antibody and this. And this is another way, another screening we use IL-4 independent screening just to see which payload is more suitable for ADC. We can see a synergy effect or additive effect. And you can see we have, in this case, antibodies have no effect without but we see several different payload have a different kind of inhibition of the CD423. So with those, we've definitely done a multiple screening. So we decide to do an in vivo model test. As you know, the in vitro is really hard to mimic the real situation in case. So we will work with the pharma. Luckily, we form with GemPharmatech. GemPharmatech have the right model. They have humanized the IL-4 and IL-4R model. So we kind of work with them to use this model and test our ADC to see whether we can see the same kind of effect we see on the in vitro. And this is the kind of another way to diagram to show that we sensitize the mouse, use OVA on the mouse back, for 7 days, then challenge the mouse back and right ear with the OVA again. And we full dosing. And also at the end of 20th day, we do the qPCR or also check the IgE factor, cytokine IgE factor, IgE level. So So for this kind of study, the one major evaluation is the clinical score. So this are the two different criteria for critical score for the back, for ear, and also the back. It's a little bit different, but the just evaluation the dryness, the bleeding or thermal and or dryness, and also edemas, which is the for the ear we measure the difference from right ear and the left ear, the thickness, and also for the back we also looking for the thickness of the back to compare to the normal mice. And this is the clinical result. We read the clinical scores on the left side is the back score, and the left side ear score. So you can see on the blue line is our ADC, and the yellow line is the antibody alpha and antibody, which is used for the ADC. And you can see we actually see quite a significant reduction of the score on the back, but on the ear it's actually kind of a bit less. It's less obvious. But we also measure the IgE level on the day 5 and the day 15. Actually, you can see on the group 4, we see quite a bit good improvement. Although it's not very significant, and in this case, it's versus group 3 and group 2. But on day 15, we also see a reduction in IgE. So it is kind of proof, our ADC does have a better or improve the thickness of the lesion of the mouse. So this is kind of like a slide I want to kind of emphasize. So we are kind of go back to see, that for ADC actually probably we can have inhibition actually beyond the alpha-R pathway. As you see here, there's in the immune system is a very complicated system. When they have an inflammation started, you probably have a multiple different cytokine can affect the disease stage. So like alpha-R can affect the alpha and alpha-13. So the antibody itself can inhibit those two pathways. But if you or the patient have a different cytokine, and there also can, like for example, like 31, you can have a different pathway. So ADC actually can not only inhibit the alpha related pathway, but also can inhibit the non-related pathway. So probably going to have a better clinical outcome. So as a summary, we have developed alpha-R to at least we did as a proof concept. We already have demonstrated we use in our cis-X conjugation method and immunomodulator. We make an ADC which is proved in the in vitro study and also in vivo study. We see a significant improvement over the antibody. But like you say, there's a reduction is not as significant in some case. So we kind of go back to look at our model, and also we see, the model that time the GemPharmatech developers only have IL-4R, humanized 4 and 4R, but don't have IL-13. So we might missing the 13 part. So the model driven maybe also by some other cytokine. So we should maybe that's the reason we'll have a new model probably going to get a better improvement. So and also we are continue screening the better payload and to improve this potency. So I want to thank you for your join us today, and really appreciate your patience here. And Jun and I will be happy to answer all your questions. Thank you.
Okay, wonderful. Thank you so much, Gong, and Jun, for those fantastic presentations, both. All right, Gong, Jun, are you ready for some questions?
Yes.
Yes.
Okay, perfect. Maybe we'll start with one for Jun. There's a couple here about some of the studies that you showed. One mentions a case study for, I'm not sure which one that was, but about the pathology in the kidney. Do you show any increased or decreased, pathology, and what general condition were the kidneys?
Yeah, so the question is about the HSC reconstituted the NCGM model, whether there's kidney pathology observed in these models. So far, we haven't observed kidney pathology in these humanized model, the reconstitution models yet. I think partly due to the reason the reconstitution is we can't really fully reconstitute the full human immune system. So the limitation of these type of models is one is donor dependent. For each HSC reconstitution, we have to use different donor. So the disease pathology can depend on the donor we use. And also, it's because it's a human immune system into the mouse. Still, we don't have enough understanding of how the human immune system interact with the mouse. Although we are able to show some of the human immune cell population, whether they can be depleted in these immune deficient mice, but I think there is limitation, especially on the later stage phenotype, for example, the kidney pathology. However, in some of our other models, we did observe clear kidney pathology, but in the humanized NCGM model, unfortunately, we were not able to see the kidney pathology. But that's a great question.
Okay, fantastic. This is a question maybe for both. Are you looking at prospective new drugs, or are you checking existing drugs with your models as well?
Yeah, I can also address this question. So typically when we build a new animal model, we would try to look for any approved drug and see whether we can use that approved drug as a proof of concept to test whether, or positive control to test whether they work well in our model. Because that increased the confidence for any new drug candidates. We are able to use that approved platform to evaluate the drug. So we always try to look for a approved drug or a drug candidates that's in the late clinical stage to prove the platform or prove the model itself. But most of the studies, I would say, for we would also try to evaluate new drugs because that's the purpose of building these animal models.
Okay, fantastic. Maybe there's a couple of somewhat related questions here for both of you. I think, do you have any mouse models for oncology studies? And perhaps there's one specifically asking about BioRay, if there's anything working with these oncology models.
I guess I can answer the question for the oncology models. So the humanized models, they not only are built for testing autoimmune disease drug candidates, but they can also work. They work great for evaluating oncology models, oncology drugs. Many of the drug targets are shared between different disease types. So a good example would be the B-cell targeting therapeutics. So I gave one example of, for example, T-cell engager targeting CD3 and CD19. So in oncology setting, people would use the same drug to treat lymphoma, B-cell lymphoma, but B-cell pathology is very, very well known in many of the autoimmune disease settings. So the same drug can actually be used for two different purposes, either oncology or autoimmune setting. So that's why we see now there are more and more B-cell targeting therapeutics that are being applied to autoimmune research.
Okay, fantastic. I suppose with the complexity of the immune system, the overlap can sometimes be helpful as well.
Yeah, I agree with Jun. So yeah, we actually have a program which is both in oncology and also immune disease. Also, to answer the question, we have a majority of our programs is the oncology project, and then we are starting doing the immuno disease project. I think it's for For the oncology project, it's actually model is much simpler. But I think the immunology project is very difficult because most it's like I mentioned, it's a multiple pathway. It's really hard to possibly get a humanized to that. So I think it's a very challenge to immunology project.
Okay. All right, fantastic. Thank you both. Some more technical, specific questions here, perhaps. Why do you have to use a double knock in mice, such as humanized TSLP and humanized, I think it's TSLP receptor, I believe.
Yeah, so I will address this question. So sometimes when we humanize a receptor, there's a reduction of binding affinity to the mouse ligand, vice versa. So the result is the signal transduction may be compromised. So in these cases, we would carefully evaluate whether there's impact on signal transduction, and eventually would make a decision to humanize both the ligand and the receptor to ensure the endogenous signals transduction is not impacted. So it is a very common strategy, not only TSLP receptor. For example, we also have the human PD-1, PD-L1, double humanized. There are many examples. That's why we keep producing double humanized, triple, even quadruple, to make sure the endogenous mouse immune cell function is not impacted.
Okay, great answer. That's very clear. Makes perfect sense. I think this question may be for Gong, but maybe both. How do you choose the payloads for the antibody-drug conjugates to treat autoimmune diseases, and how do you approach it differently for oncology, say?
Yeah, that's a very good question, and also a very challenging question. So, I think it's for oncology actually it's much easier because you just kill the cell, right? You can multiple ways. You can use a tubulin inhibitor, topo I, and also, but you don't concern most of your concern is a side effect, which is not affect other cell. But the autoimmune disease is much difficult because you don't want to kill the cell, right? Because they have other different. So you need to have a more better understanding of the immunology and the biology underlying this disease model. So I would say you need to try I think it's good things that there's a lot of different array of small molecule for they already have in the market. So and also the target that you have. So you can choose, but the key is just don't kill the cell, and and also to try not to introduce some other effect. Like for example, like ADC for the oncology, you want a bystander effect, but for the autoimmune disease, you probably want to opposite the way. You don't want it to bystander effect, which you want a very clear, faster clearance of the payload once they leave your target. Yeah.
So a question of clearer endpoints and careful consideration. It makes sense given the mechanisms. Wonderful. What are the limitations of, for example, humanized HSC, NCG, M-based autoimmune models?
Yeah, so a few limitations. So one is for each donor cell, we can only reconstitute a certain number of mice. And also because the phenotype sometimes depend on the donor, so we may expect to see a donor-to-donor variation in terms of the disease phenotype, and the response to treatment. There's also increased variation compared to some easier model using, for example, the immunocompetent models are usually more consistent because they don't rely on donor cells.
Okay, fantastic. There's a question here about the anti-TNF antibody-drug conjugate. Can it be used in reference to tumors? Is it only used for specific tumor subtypes, operable tumors, etc., or is it broader?
If I understand right, so the question is whether use anti-TNF antibody, ADC, to treat the tumor, right? Because it is anti-inflammatory.
Exactly.
Like I mentioned before, TNF itself is not really good. To me, I think it's not good target.
Right.
If it's possible, I'll say they're using TNF-R1 or R2, especially for R1. And maybe that's more related to the inflammatory. Probably use that TNF-R1 as an ADC target. Probably better than that way.
Is that due to sort of specificity and selectivity then, Gone?
Yeah, I think it's based on the internalization. Because the
Internalization.
TNF is the cytokine. Although you can use a membrane bound TNF, but majority is still soluble. I mean, in the solution, so it's not really good way to bring the payload to the inside the cell, the tumor cell.
Okay, fantastic. We're getting near the hour mark here, but I think we have time to squeeze in at least a couple more questions. One here is, what is the process for validating human gene knock-in models?
So whenever we build a new human gene knocking model, we would typically check for the transgene expression. We wanted to make sure the human gene is expressed correctly, whether they are in the correct tissue or cells, or if they're expressed at the right level. These are the first step of QC. Then we would put these humanized mice in a disease setting, for example, a autoimmune disease or oncology model or a metabolic model, depending on the context, and to prove whether these humanized model can be utilized for evaluating drugs in these disease settings. So yeah, both expression level and the functional level validation we try to implement for each model we generated.
Okay, fantastic. Just a reminder too, we have in the handouts a number of resources and links to some of the, the different models and things that are being discussed today. So please do check those out. A question for both of you, I think probably here. What are some of the limitations of antibody-drug conjugates?
So the limitation. It was a great question. So I think the whole industry working on this, right?
Right.
I think it's so the limitation for oncology, and it's very different from the immune disease one. Oncology, like I mentioned, is as far as you have the select target and also the most potent the payload. It's probably can do good, but for the immune related disease, then because all the cells you targeted the immune cell, which could be expressed, and the target could be expressed on all different immune cells. So, and then how do you choose the right target, and also how do you choose the payload to really selectively to the particular pathway, and that's the really need to fully understand the biology of those. I think that's a challenge part.
Yeah, I think it's especially because ADC is a very complex drug. It has the antibody arm and linker and the payload. So you have to consider all of the three and consider the interaction between each pair. And so that just increase your variability. So it requires a lot of research and understanding of the biology. If one part is wrong, then your whole ADC molecule may be wrong. So it's a very complex drug.
Yeah, that makes sense. Just follow up on the Jun. So, for us, actually, most challenges is find the right testing in vitro, because it's really hard to mimic in the real situation, and also really hard to find an in vivo model. We're lucky, like working with the GemPharmatech to find some humanized model. But still, that like if we do a double and triple, even more quadruple, like replacement, that's probably more in the real situation, in the disease.
Yeah, and the other autoimmune diseases are very complex, and often the pathogenesis of the disease is not very understood. So hopefully, with the whole world working on these models and developing innovative medicine, we are able to advance the treatment for patients.
Wonderful. I think that's maybe we'll squeeze in one last just sort of reference question, because I think that's a great point to leave off on. Are there other applications of ADCs for non-oncology, non-autoimmune disease indications, or perhaps is there somewhere we can point someone with that information?
That's a good question. So I think it's ADC is almost like antibody combined with a small molecule. So it's just, you can think of it as those antibody field and also the small molecule drug field. All can think about. One thing that I think is beside the oncology and immuno disease, it's one actually Genentech, they actually done some testing using the anti-infected disease. So to target the bacteria and carry the antibiotics, so then kill the selectively bacteria. So especially right now, there's more, resistant bacterial strain come out. Maybe that's one field, but you need to find the right target, which can bring into the back. And maybe also, I think I know that we collaborated before with some company, the hospital. They're doing virus one, which is like some virus. It also exposes like HIV, they expose on the protein on the surface. So you can bring the ADC to kill those virus infected the cell. So it's kind of trying to prevent the proliferation of the virus. Yeah, I think it's just the field actually kind of expanding right now. Yeah.
Wonderful. Okay, unfortunately, I think that is all the time we have for today. But a huge thank you to Jun and Gong, to both of you for joining us today. The fantastic presentations and sharing all your insights. It's been a pleasure having you with us.
Thank you.
Thank you, Andrew.
Thank you, everyone.
All right. And of course, yes, we would certainly like to say a big thank you to our audience for participating here today. We know you have very busy schedules and certainly appreciate you joining us. And in closing, we hope you enjoyed the Scientist.com and GemPharmatech webinar, and we'll see you again next time. Thank you, everyone.